Good morning friends. Today we will discuss about direct ELISA. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Direct ELISA is a type of enzyme-linked immunosorbent assay. This technique is used to detect as well as measure antigen by using enzyme-level antibody. Direct ELISA is simpler and faster as it has fewer steps. It is used in clinical diagnostics and research. Different steps of direct ELISA in a sequence are immobilization, blocking, antibody incubation, washing and substrate addition and detection. First step in direct ELISA is the immobilization of antigens at the bottom of the wells of a microtiter plate. These are the antigens that are immobilized at the bottom of the wells. For this, a sample, for example, patient serum suspected to have the antigen is added to the well and then incubated for a period of time. In direct ELISA, antigens are immobilized by direct adsorption at the bottom of the wells. After immobilization of antigens, the plates are blocked by adding proteins such as bovine serum albumin or ovalbumin. Blocking is necessary to ensure that the enzyme-linked antibody binds only to the target antigen. After the plates are blocked, next step is antibody incubation. For this, a solution containing a specific amount of enzyme-linked antibody is added to the wells and then incubated for a period of time. These antibodies are linked with enzymes such as alkaline phosphatase or horseradish peroxidase. So now the antibodies will bind to the target antigen and as a result antigen-antibody complex will be formed. So these are the enzyme-linked antibody which binds to the target antigen that are immobilized at the bottom of the wells. As a result of this binding, antigen-antibody complex will be formed. Because the concentration of enzyme-linked antibody is higher than the concentration of antigen during incubation, some antibodies will bind to the immobilized antigens and remaining will become free. So these are the antibodies that remain free or unbound. Such free or unbound antibodies are then removed from the wells by washing the plate with phosphate buffer saline. Now the question is, what will happen if these unbound antibodies are not removed? The answer is that in this case, when a chromogenic substrate is added to the well, the substrate will be converted into product by the enzymes which is linked to the bound antibodies as well as by the enzyme that is linked to the free or unbound antibodies. As a result, more amount of product will be formed and a higher value will be obtained. So in order to get accurate result, all such unbound antibodies are removed from the wells by washing the plate with phosphate saline. After all unbound antibodies are washed off, a chromogenic substrate is added to the well. These enzymes that are linked with the bound antibodies will convert the chromogenic substrate into a color product. This enzyme will convert a chromogenic substrate into a color product. The amount of this product is proportional to the concentration of antigen in the sample. Now the question is how to determine the concentration of sample antigen. In order to determine the concentration of sample antigen, a standard calibration curve is prepared. For this, a series of standard solutions having increasing known concentration of antigen are placed in different wells of a microtiter plate. So these are the wells of a microtiter plate. In all these wells, a series of standard solution having increasing known concentration of antigen has been placed. In next step, a specific amount of enzyme-linked antibody is added to all these wells and then incubated. This is followed by the addition of a chromogenic substrate. At the same time, the sample which has already been subjected to ELISA protocol is placed in another well of the same plate. After development of colors, the optical density of standard solution and that of sample is determined by a plate reader. After determination of optical density, a standard curve is plotted by taking concentration of standard solution on x-axis and their optical density on the y-axis. From this standard calibration curve, the concentration of sample antigen can be easily determined. Direct ELISA has several applications. It is used for screening biological samples for the presence of antigen. In addition, it is also used for detecting food allergens and also for tracking disease outbreak. It has several advantages. Number one, direct ELISA is simpler and faster. This is because it has fewer steps and it requires only one enzyme for detection. In addition, in this technique, the background noise is also reduced. However, this technique also has several disadvantages such as lower sensitivity, limited flexibility, and in addition, there is also a potential for non-specific binding. Friends, this is all about direct ELISA. For more updates, subscribe my YouTube channel, Sam Biology Class.